Hello, everyone. I am Yogi, an associate professor of the Nutrigenomics Research Group. Today, I will talk to you about nutrigenomics, introducing our recent progress in this research field. This is a photo of the members of my research team, Nutrigenomics Research Group. The Nutrigenomics Research Group is based on the Laboratory of Molecular Physiology on Energy Metabolism, which was established in Tokyo University Graduate School of Medicine from 2008 to 2011, and since then we have been working as a research group spanning the University of Tsukuba and the University of Tokyo. So, today's topic is nutrigenomics. Nutrigenomics is a research field of studying how a nutritional environment affects gene expression on a whole body scale. Nutrients first act on the genome. That leads to changes in gene expression, and that in turn changes the metabolism of nutrients. Nutrigenomics deals with this whole system and can be described as a kind of systems biology. Here's a question: what are nutrients? Let me briefly mention this point. The body's energy needs are mainly supplied by the three major nutrients: protein, fat, and carbohydrates. These are also referred to as macronutrients. Other nutrients include vitamins and minerals, and sometimes also include fibers and plant-derived chemicals called phytochemicals. Today's lecture will deal primarily with macronutrients. This is today's contents. First, I will introduce the concept of nutrients as information carriers. Next, we'll look specifically at macronutrients and how they act as information carriers. In the last part, I will talk about nutritional signals during fasting. So, first I will talk about the concept of nutrients as information carriers. Recently, I edited a textbook of nutrigenomics, and published as a series of Jikanagaku. Although this book is written in Japanese, please try to read it if you have a chance. In this book entitled Nutritional Signaling, a new key player in gene regulation, we describe the aspect of nutrients as information carriers with some examples of nutritional signaling. The next slide is cited from this textbook. Here are some examples of nutrient signals. For example, regarding glucose metabolism, it is known that alpha Kg, an intermediate metabolite of the TCA cycle, is used as a substrate for histone and DNA demethylases involved in the regulation of epigenetic information. The recent discovery of new sensor molecules for some amino acids, such as cystine and castor, is also noteworthy. Metabolites related to lipid metabolism such as butyrate are also involved in the regulation of epigenetic information. So, let's return to today's list of themes. In the first part, I introduced the concept of nutrients as information carriers. Then, we'll look specifically at macronutrients and how they act as information carriers. The first topic is about glucose and its related metabolites. This slide shows the various regulatory mechanisms of gluconeogenesis. Gluconeogenesis is a metabolic pathway that results in the generation of glucose from certain non-carbohydrate carbon substrates. This example shows that besides hormones such as insulin, glucagon, and glucocorticoids. Some metabolites involved in the glucose metabolism, such as acetyl-CoA and NAD+, are also controlling gluconeogenesis. And the next topic is about amino acids as signaling molecules. Recently, new amino acid sensors have been discovered and are attracting much attention. GCN2, or General Control Non-Irrepressible 2, is a serine threonine protein kinase that senses amino acid deficiency through binding to uncharged transfer RNA. In protein synthesis, no amino acid compensates for the absence of another. Therefore, the cell must be able to efficiently detect the lack of any amino acid to prevent potential failures in peptide chain synthesis. The GCN2 system contributes to this detection. Under low intracellular amino acid levels, the binding of GCN2 to a given uncharged transfer RNA triggers a conformational change that leads to kinase activation and inhibitory phosphorylation of a key early activator of translation initiation, EIF2 alpha, or eukaryotic translation initiator factor 2 alpha. This slide shows how this GCN2 system works to control FGF21 expression in response to low-protein diet. FGF21, or fibroblast growth factor 21, is a circulating hormone associated with metabolic responses to nutrient restriction, and FGF21 deficient mice fail to exhibit the increases in food intake, increases in energy expenditure and reductions in growth observed in wild-type mice consuming low-protein diet. As shown here, this paper shows there are two pathways, GCN2-dependent and GCN2-independent. They showed that FGF21 knockout mice are fully resistant to low-protein-induced changes in food intake, energy expenditure, body weight gain, and metabolic gene expression, and GCN2 knockout mice recapitulated this phenotype for a short period, but subsequently, low-protein-induced effects on food intake, energy expenditure and body weight begin to appear after 14 days on diet via a GCN2 independent pathway. Recently, new amino acid sensors have been discovered and are attracting attention. They are Castor-1 and SLC38A9 for arginine and Cystrin-2 for leucine, respectively. Both are connected to mTOR1, or mammalian target of rapamycin complex 1, a protein complex that functions as a nutrient energy sensor and controls protein synthesis. mTOR signaling mainly controls mRNA translation and protein turnover, but also regulates glucose, lipid and nucleotide metabolism. And in the regulation of metabolism, some transcription factors such as ATF4 and TFEB are involved. Thus, information on amino acid is conveyed to nucleus via mTOR. And the next topic is about fatty acids as signaling molecules. As I mentioned earlier, including small intermediate metabolites derived from fatty acids, there are so many signaling pathways related to fatty acids. In this part, I'd like to focus on long-chain fatty acids. Consumption of specific dietary fatty acids has been shown to influence risk and progression of several chronic diseases, such as cardiovascular disease, obesity, cancer, and arthritis. In recent years, Insights into the mechanisms underlying the biological effects of fatty acids have improved considerably and have provided the foundation for the emerging concept of fatty acid sensing, which can be interpreted as the property of fatty acids to influence biological processes by serving as signaling molecules. An important mechanism of fatty acid sensing is via stimulation or inhibition of DNA transcription. In these regulations, fatty acids are sensed via such receptors as PPAR, or peroxisome proliferator activated receptors, toll-like receptor 4, G protein-coupled receptors, and other putative mediators. The PPARs perhaps compose the best recognized sensor system for fatty acids. PPARs are transcription factors that are members of the superfamily of nuclear hormone receptors, which also include receptors for fat-soluble vitamins A and D and steroid hormones. Nuclear receptors function as ligand-activated transcription factors by binding small lipophilic molecules. They share a modular structure consisting of a DNA and ligand binding domain and play a role in numerous biological processes. 3 different PPAR subtypes have been cloned, each characterized by a unique tissue expression pattern. PPAR alpha, which regulates a number of genes involved in beta oxidation of fatty acids, is found in many tissues but is predominant in oxidative tissues such as brown adipose tissue, cardiac muscle, skeletal muscle, and liver. PPAR delta is found in many cell types, whereas PPAR gamma expression is more restricted, with the adipocytes and macrophages expressing the highest level. PPAR gamma is also known as the master regulator of adipocyte differentiation. Binding of ligand is thought to trigger the physical association of PPARs to specific DNA sequences, called PPAR response elements, in and around target genes. PPARs also act as repressors against other transcription factors. In several landmark articles from the 1990s, it was demonstrated that all 3 PPARs are able to bind fatty acids with a general preference for PUFAs, or poly-unsaturated fatty acids, although other fatty acids can also bind to PPARs with lower affinities. PUFAs are fatty acids that contain more than one double bond in their backbone, and as you can see, they are strongly flexed. From here, I would like to focus on PUFAs and explain how they work in a little more detail. Since the 1960s, PUFAs have been known to inhibit the synthesis of fatty acids in the liver. As you can see here, only linolete suppresses hepatic lipogenesis, while other fatty acids such as palmitate and olete do not. Linolete is a PUFA expressed as C18, 2, which means it contains 18 carbons and 2 unsaturated double bonds. This inhibitory effect of PUFA on hepatic lipogenesis is also observed in PPAR knockout mice, and therefore, this is thought to be due to a PPAR-independent mechanism. Shown here is a schematic of the metabolic pathway of de novo lipogenesis. De novo lipogenesis is a metabolic pathway that synthesizes triglycerides from glucose. Acetyl-CoA is a hub metabolite on this pathway, which can be used as the starting material for both cholesterol and fatty acid synthesis. As mentioned earlier, PUFAs suppress hepatic lipogenesis, and in the 1990s, it was found that this occurs at the transcription level. As shown here, gene expressions of a series of lipogenic enzymes are suppressed in concert by PUFAs. Lipogenic enzymes are enzymes involved in fatty acid and triglyceride synthesis. They also include those involved in the production of NADPH, because NADPH, instead of NADH, is used as a cofactor in the lipogenic reaction pathway. They are strongly induced in the liver and adipose tissue of red-fed animals. Conversely, they are suppressed when the animals are fasted. And they are primarily regulated at the transcription level. In the liver, they are controlled by a transcription factor SREBP-1, or sterile regulatory element binding protein 1. SREBPs are transcription factors that belong to the basic helix loop helix-leucine zipper family, and are considered to be controlling the transcriptional regulation of triglyceride and cholesterol synthesis. As shown here, they bind to DNA as a homodimer. From here, I would like to proceed based on our own research. When SREBP-1 is overexpressed by a transgene in a transgenic mouse model, downstream lipogenic genes such as FAS and ACL are upregulated in the liver. This leads to marked elevation of hepatic triglyceride content, that is, fatty liver. Conversely, SREBP-1 knockout mice show very small responses of lipogenic genes when animals are refed. Plasma Tg levels are also lower in SREBP-1 knockout mice. From these lines of evidence, we hypothesize that SREBP-1 is involved in the suppressive effect of PUFAs on lipogenic enzyme gene expression, and tested the hypothesis. As shown here, PUFAs actually reduced the amount of SREBP-1 in the liver nucleus. In contrast, SREBP-2, which is mainly involved in the regulation of cholesterol synthesis, was not affected by PUFAs. Next, we tested whether the protein expressed by the transgene is affected by PUFAs. As shown in the right panel, the amount of SREBP-1 protein derived from the transgene was not reduced by PUFAs. The difference between endogenous and transgenic SREBP-1 is its length. The transgenic SREBP-1 used here is artificially shortened and lacking in membrane binding and regulatory domains. Moreover, in order to clarify whether the effects of PUFAs are totally mediated by SREBP-1 or not, we tested the effects of PUFAs on the SREBP-1 transgenic mouse liver. The left panel shows the effects of PUFAs on wild-type animals, and the area circled by the red square shows the suppression by PUFAs. By contrast, as shown in the right panel, the effects of PUFAs are completely abolished in SREBP-1 transgenic animals. This result demonstrates that the effects of PUFAs are totally mediated by SREBP-1. In the previous slide, I mentioned the difference in length between endogenous and transgenic SREBP-1. Actually, SREBP-1 is initially made as a ER membrane-binding precursor form, and then it is cleaved enzymatically into the active form, which migrates to the nucleus. The transgene is made of the short active form. The fact that the transgenic SREBP-1 was not affected by PUFAs suggests that PUFAs suppress the cleavage process without affecting the stability of the cleaved protein. In order to clarify if PUFAs suppress the cleavage process, we planned another experiment. In this experiment, we used a reporter system utilizing the yeast transcription factor GAL4. GAL4 recognizes genes with UAS, an upstream activating sequence, and activates them. Therefore, we attached GAL4 and VP16 activation domain to the N-terminus of SREBP-1, and constructed an assay system to detect the GAL4 signal using an UAS-driven luciferase reporter. Using this reporter system, we found that PUFAs specifically suppress the SREBP1 cleavage, while SREBP2 is not affected as shown here. EPA stands for eicosapentaenoic acid, a kind of PUFA. The mechanism by which PUFAs suppress the SREBP1 cleavage is still unknown, and we are continuing to investigate this point.